Good afternoon to all. This is Barathi from Microbiology Department. Now I am going to tell you about antimicrobial resistance. By seeing the picture itself, you can see what is antimicrobial resistance. It means it is a term related to microorganisms. We know that most of the diseases in the world for human beings as well as animals are caused by variety of microorganisms. So those microorganisms, so what they are telling the microorganisms, some microorganisms are telling, I'm scared of antibiotics, but some microorganisms, they are telling, no, don't worry, no need to scare about antibiotics. Okay. So there are two types of microorganisms in general, that is antimicrobial resistant organisms, and the another one category which is sensitive to the antimicrobial treatment. Okay, so from the antimicrobial resistant organisms, the antimicrobial sensitive organisms will be gaining the resistance power. So I am going to explain you now about that. So here a brief introduction. So antimicrobial agents or treatment providing agents which are introduced recently, that is in the year of 1929. Actually penicillin, that is called queen of medicine. This penicillin was introduced by that was accidentally discovered by Alexander Fleming and was introduced in the treatment of some disease treatment. Okay, but as soon as the years gone, the organisms gained the resistance against the particular antibiotic. So initially, before penicillin, one antibiotic, one chemical component was used as an antimicrobial agent, that is known as sulfonamide. After that, only penicillin was used frequently. That is why the organisms gained resistance. So likewise, there are different types of antibiotics had been introduced by different scientists from different countries, but whatever the organisms, all types of organisms gain resistance against those microorganisms. So here in this slide, the antimicrobial agents, in general we used to say antibiotics, and those antibiotics provides protection against microorganisms for human beings as well as animals. So here the antimicrobial resistance is nothing but the ability of a microbe to resist the effects of medication which was previously used to treat them. Okay, so antimicrobial resistance is nothing but it is a protective mechanism exhibited by microorganisms. So here, this antimicrobial resistance, what it do, it is responsible for millions of deaths worldwide, and it is considered as one of the major health concern nowadays. Then this antimicrobial resistance threatens the effective prevention and treatment of an ever increasing range of infections caused by a variety of microorganisms. And the cost of health care for patients with resistant infections is higher. Because of these reasons, the antimicrobial resistance is prevalent. So this slide shows you how the antimicrobial resistance had been occurred in worldwide. See here, two ways available. One is through the animals and another one through human being. How the antimicrobial resistance developed among other microorganisms. So now we go for how the antimicrobial resistance, in brief we used to say AMR. The AMR had been occurred from animals. So here variety of animals have been given, a chick, a cattle and pig. So here the animals get antibiotics and develop resistant bacteria in their gut region. Then when these animals or the product from these animals used for some other purposes, the antimicrobial resistance can be spread. For example, fertilizer or water containing animal feces and drug resistant bacteria is used on food crops. So we know that the fertilizer or animal feces can be used as a fertilizer for the cultivation of crop. In some regions they used to do that. So because of that, the antimicrobial resistant bacteria from the gut of this animal may come to the earth, and that is may come to outside, and it may be responsible for the spreading of antimicrobial resistance. Then the drug resistant bacteria in the animal feces can remain on crops and be eaten. These bacteria can remain in the human gut. So from the animal gut to human gut, the bacteria transferred. So thus responsible for antimicrobial resistance, and then automatically it will be present in human being. So the human will get the antimicrobial resistance. So this is one of the method. And one more method, the drug resistant bacteria can remain on meat which is available from animals, and when not handled or cooked properly, the bacteria can spread to humans. So by means of two ways, the antimicrobial resistant organisms can be entering into human beings. One is from the animal feces, it can be used as a fertilizer for the cultivation of crops, and another one by eating the improper cooking of meat which is obtained from animals. So because of these reasons, the antimicrobial resistance is prevalent. And how the antimicrobial resistance can spread by human being means, imagine this fellow named something, and this fellow got some antimicrobial resistance, some infection, and for curing that infection he got an antibiotic. He has taken an antibiotic, but he is having antimicrobial resistant organism. So now you see this fellow, how he is spreading the antimicrobial resistance means by means of variety of methods. So first one, for treating the infection, this person goes to hospital or some healthcare center. Okay, so from there what will happen? He is spreading the antimicrobial resistant organisms. How means? He is walking in the corridor and he is sitting in the chair. Through his sweats or through coughing or sneezing, somehow he is spreading the microorganism. Then the person, the healthcare person who are handling this person may be indirectly spreading the antimicrobial resistance to others. Okay. Now these healthcare patients or other patients, those who are available in the healthcare center, okay, after treatment got over, the patients will be going home, and at home these patients may be responsible for the spreading of those antimicrobial bacteria to others. So as a whole family, these people will be getting antimicrobial resistance. Okay. So by means of different methods, the antimicrobial resistance can be spread. Then this is the timeline of antimicrobial resistance. So I mentioned previously, that is in the year of 1930 itself, an antibiotic, we used to say a chemical that is called sulfonamides, was introduced. Within 10 years, that is at 1940, many microorganisms developed the resistance mechanism against sulfonamides. So immediately the scientists have done many research work and introduced one more antibiotic, new antibiotic, that is penicillin. So within 5 years, the organisms got resistance against penicillin. So again the scientists have gone for one more alternate, that is called another antibiotic, streptomycin, chloramphenicol, tetracycline. So if one antibiotic, scientist develops one antibiotic, within 5 years, the organism, not even 5 years, within 2 to 3 years, the organisms used to develop resistant mechanism. So there is a need for the invention of new antibiotics, discovery of new antibiotics, because organisms used to develop their resistance mechanisms. Okay, so this is the timeline which shows the antimicrobial resistance emergence from 1930 to 2005. Then factors. Okay, so we know what is antimicrobial resistance. Now we go for what are the factors which are responsible for antimicrobial resistance. So here I have given 4 points which are mainly responsible for antimicrobial resistance. The first one, drug related factors, and second, environmental factors, third, prescriber related factors, and fourth, patient related factors. Then, so what is meant by drug related factors? So in the forthcoming slides we used to see. See, this is the definition for antimicrobial resistance. We have seen already, and this is what are the factors. So coming to the environmental factors. The first one, huge population and overcrowding, like cinema, park, beach, railway station, wherever you go, malls, big malls, wherever you go, there are huge populations and overcrowding available. So in there, among those, some people will be there. Many people will be having variety of infections, and they used to take variety of medications, okay, which includes different antibiotics. So from those persons, infected persons, we may get antimicrobial resistance. So huge population and overcrowding is one of the factor which is responsible for antimicrobial resistance. And this is we used to say advanced one because now we have well developed transport facilities, but even though that is one of the main drawback of the spreading of antimicrobial resistance, because we used to go here and there, okay. So because of that, the antimicrobial resistant bacteria can be spread. So rapid spread due to better transport facilities and poor sanitation. It means cleanliness, okay. So the cleanliness will be very low, that is poor sanitation, and increase in community acquired resistance. So community acquired resistance, in the sense, community means group. So in group the antimicrobial resistance will be developed. That is called community acquired resistance. So these altogether represents environmental factors for antimicrobial resistance. And what are the other factors, in the sense, ineffective infection control program. See, there are different governments, they will be implementing different strategies for the control of a particular infection or variety of infections. So if those control programs are not effective, in the sense, the antimicrobial resistance will be prevalent. So ineffective infection control program, that is one of the main factor. And widespread use of antibiotics, especially in animal husbandry and agricultural, and as medicated cleansing product. It means if you go to a veterinary hospital, normally human beings, they used to take antibiotics without any medication, without doctor's advice. They used to take, if they have fever or if they have throat infection, they will go to the medical shop and they will get one azithromycin or erythromycin, something. They will take an antibiotic, okay, but they won't be taking the proper dose of antibiotic. If they take one tablet, they feel okay, then they will stop taking antibiotics. That is called improper use, okay. So because of that, the antimicrobial resistance is prevalent, right? And here in animal husbandry, that is animal cultivating and animal processing, that we can say, in animal husbandry, variety of antibiotics will be used, because animals are prone to different kinds of infections. Most of the bacterial infections and most of the viral infections will be occurring in animals, and from the animals there is a possibility for transferring to human beings, okay, that is called the zoonotic diseases. So to treat the diseases in animals, there are variety of antibiotics have been given. And in the field of agriculture, for crop cultivation, to delete or to destroy variety of microorganisms, some kinds of antibiotics will be used. So in animal husbandry field and agricultural field and cleansing material, preparation of cleaning materials, the antibiotics will be added. So because of that, so what we will be, we are exposing to the more quantity of antibiotics. Frequently we are exposing. See, if we are wiping our home, what we used to do, we will take off bucket water, then put three slots of phenol, that's it. Then we used to wipe. But in hospitals or in animal husbandry or in agriculture, what they used to do, they will add some antibiotic. So without knowing or unknowingly or indirectly, we are exposing to the antibiotics. So because of that, the antimicrobial resistance is prevalent. And release of large quantities of antibiotics in the environment during manufacturing. From the pharmaceutical industry, during manufacturing, many antibiotics will be released as a waste material. So usually the wastewater treatment system is located, is built in all kinds of industries, tannery, textile, leather and fertilizer industry, paper industry, and almost all the industries wastewater treatment system will be there. In that case, if the wastewater is not treated properly, the antibiotic is discharged into the environment. So in that particular area, who are all present in that area, they will be exposed to the antibiotic, and there is a possibility of occurrence of antimicrobial resistance. So by means of variety of ways, the antimicrobial resistance had been occurred. And coming to the patient related factors, here the patient may be having poor adherence to the dosage regimen. So the doctor used to say you have to take 500 milligram, and better you can go for 750 milligram, like that. So that is called dosage regimen. Okay. So some patients, they cannot adhere or they cannot take that dosage because they feel so irritation, bowel irritation, or they will be having severe stomach pain, like that, vomiting, nausea, everything will be there. Okay, so poor adherence of dosage regimen is one of the main factor. And poverty in the sense, in Tamil, varumai. So because of poverty, the patient is unable to buy the medicine. Okay, so there is no frequent take of medicine, or correct dosage of medicine is not taken, in the sense the patient may show antibiotic resistance. And lack of sanitation concept, that is cleanliness, there is no cleanliness, and lack of education. This much we have to take, no proper awareness will be there. What the patient, if one patient is coming from village, in the sense, what they used to say, no need to take this kind of medicine, this will affect your stomach very badly, and you will be getting wounds in your mouth, so don't take medicine and all, like that they used to say. So that is called lack of education. So we should give proper education. And self-medication. In city side, what the people will do? Okay, for throat infection we can take azithromycin 500 mg for 2 days, for 1 day, morning and night we can take, then the cold will be subsided. Like that the people will be thinking, those who are present in city side. So that is called self-medication. Okay, not only in city side, of course in all the area, if we are taking medicine self, that is called self-medication. So it should be avoided compulsorily. And the one more thing, that is prescriber related factors. So here the inappropriate use of antibiotics, that is the doctor used to say you have to take medicine for 3 days, that is for 6 times you have to take, morning, night, like that you have to take. But what we do, we won't do like that. No, I feel stomach burning, I feel stomach ache, vomiting. So we say some reasons to avoid, for avoiding to take the medicine. So what will happen? Automatically we are not taking the proper dosage, that is called inappropriate use of antibiotic. Otherwise we won't take correct medicine. So, see, in case of self-medication, without doctor advice we are taking. So in that case we are not taking proper antibiotic. So that is called inappropriate use of antibiotic. And increased empiric antimicrobial use. Poly means many. So instead of taking only one antibiotic, we can go for combination of antibiotics. Azithromycin, erythromycin and streptomycin. These three will be taken altogether. That is called polyantimicrobial use. And overuse of antimicrobials. So doctor used to say you can take only for 3 days. But what we will be doing? No, we can take for 1 more day. So extra one day you will be taking, that is called overuse, or frequently taking, that is called overuse, and prolonged use. See, of course, these are all will give you similar meaning only. Overuse, prolonged use. Prolonged use means for long time you are using, that is called prolonged use. And inadequate dosing. Inadequate in the sense, if you want to take 500 mg, you will be taking only 250 mg or 300 mg. So 300 mg in the sense, you should go for frequent taking. Okay, 300, 300, 300 like that you have to take. So per day, 3 times you have to take. That is called either inadequate or prolonged use of antimicrobials. And lack of current knowledge and training. So this is lack of education, which includes lack of education. Then, so this is the mechanism how the antimicrobial resistance may arise. In human region or in human gut or animal gut, whatever, the first, here the antimicrobial resistance is occurring in 4 stages. First, this gut region shows you variety of microorganisms, especially coliforms, present in the gut region, and these 2 are antimicrobial resistant bacteria. And coming to the second one, here the patient is or the person is taking antibiotic. The antibiotic will be killing the pathogen as well as some normal microorganisms, but it did not disturb the resistant bacteria. So what happens? The next stage, the normal cells, infected cells all got destroyed, whereas the resistant bacterium left over remaining. Now it is allowed to grow and take over. So this resistant bacterium will be growing abundantly, and automatically what will happen? This bacteria develops and spread the resistance to other normal cells. Okay. So through this is a simple way shows you how the antimicrobial resistance can be spread. Then here, what are the other methods by which the AMR, that is antimicrobial resistance, can be spreading? It is of 2 methods. One is intrinsic and another one is extrinsic. Intrinsic, it is also known as natural methods, and extrinsic, or we used to say acquired infection. Acquired means during lifetime we can receive that. That is so. Here, that is intrinsic or natural in the sense that includes variety of methods, and acquired, which includes variety of methods. The very famous one is genetic methods. So in that, the very famous one is mutation. Okay. So there are variety of factors which involves in the cause of antimicrobial resistance or in the spreading of antimicrobial resistance. Plasmids, transposons, integrons, and mutation. These are some of the methods, and some biochemical mechanisms also involving in the spread of antimicrobial resistance. So this is very important. So here production of antibiotic inactivating enzymes, that is the main thing through which many microorganisms exhibit resistance, and these are the remaining methods. So these are all very important. And preventing drug accumulation within bacterium, modifying or protecting target site, then use of alternative pathways for metabolism or growth, and quorum sensing. Quorum sensing is nothing but bacterial communication. Bacterial cells will be communicating one to another, that is called quorum sensing. Then extra chromosomal methods. So what we have seen in the previous slide, that is called the chromosomal methods. So here extra chromosomal methods through which the antimicrobial resistance can be spreading. So here one agent called plasmid, which is mainly involving. So plasmids, you know well, that is plasmids are antimicrobial, plasmids are extra chromosomal DNA molecules which act as DNA molecules which replicates autonomously, and through the plasmid the antimicrobial resistance can be spread. Okay, so there are some more mechanisms available: conjugation, transduction and transformation. These are some of the methods. How the plasmids are responsible for antimicrobial resistance means they possess a genes called the resistance genes. So because of that, the AMR can be spreaded among microorganisms. So this is the diagrammatic representation of antimicrobial resistance occurring in microorganisms. So this is bacterial DNA, these are plasmids. Through cell replication, when the bacterial DNA, main DNA replicates, the bacterial plasmids also replicates. If it possesses the antimicrobial resistant genes, it is also can be replicated or spreaded to another bacterial cells. Then through transposons. Transposons are nothing but jumping genes, very famous one, jumping genes which are present in DNA molecules. When the transposon possesses resistant genes and jumping from one location to another location, the organism may get resistant genes for particular antibiotic. So here some of the organisms like Staphylococci and Enterococci, they possess antimicrobial resistance genes. And certain organisms, some organisms, they used to synthesize some enzymes. So through the enzymes, the antimicrobial resistance can be expressed in microorganisms. So here some examples have been given. For example, Staphylococcus, Neisseria and Haemophilus. These are some of the organisms. These organisms produce an enzyme called beta-lactamase. Beta-lactamase. So this enzyme responsible for the inactivation of an antibiotic called penicillin. And there are certain organisms, we used to say gram-positive and gram-negative. Coming to the gram-negative organisms and the gram-positive, some of the gram-positive and gram-negative, they inactivate chloramphenicol. It is an antibiotic by means of an enzyme called chloramphenicol acetyltransferase. So it is very easy to remember. That is, acetyltransferase is the general one. If it is called the chloramphenicol acetyltransferase, it means the organism is resistant to chloramphenicol. It possesses the enzyme chloramphenicol acetyltransferase which inactivates chloramphenicol. And there are certain organisms which produces acetyltransferases or phosphotransferases and adenyl transferases. So these are plasmid or transposon mediated enzymes which are responsible for antimicrobial resistance. And this is the diagram which shows you the antimicrobial resistance exhibited by microorganisms in a clear manner. See, in biochemical mechanism, I told you inactivation or modifying of an enzyme activity or modifying antibiotic binding site, everything. So this picture represents all the biochemical mechanisms altogether. See, it's a very good. See, it is a bacterial cell. This yellow color is a bacterial cell. This is the main chromosome. Here what is happening? The antibiotic, this blue color indicates antibiotics. The blue color, that is an antibiotic which enters inside the bacterial cell. So here what is happening? The activation of drug efflux pumps by the microorganism responsible for antimicrobial resistance. Efflux in the sense removal. In Tamil, velitalludal. Okay. So the efflux pumps will be activated by the microorganism. Because of that, the drug will be expelled outside. So the organism will be escaping and surviving. And inhibition of drug uptake. There are certain organisms which inhibit the uptake of drug. So there is no drug entered inside the organism. Because of that, the organism gains antimicrobial resistance. And the binding site, how the inhibition can be expressed means through the modified cell binding sites and through the presence of plasmid the organism can express antimicrobial resistance. And alteration of drug target. See, this is the binding agent which binds with antibiotic. So here what will happen, when antibiotic enters inside the organism, the bacteria will change the site. So there is no proper binding site for the antibiotic. So what will happen, there is alteration in the site, structure of the site. That is why the antibiotic unable to bind with the binding site. So the organism will be escaping. It develops the antimicrobial resistance. And so by drug inactivating enzyme, I told you in the previous slide, acetyltransferases, phosphotransferases, so many enzymes, beta-lactamase. So these enzymes will be inactivating the antibiotic. So by means of variety of mechanisms, the antibiotic exhibits antimicrobial resistance. Then, so these are the explanation what I have told you in the previous slide. In this slide, zoom out. So what I have shown you in the previous slide has been given in words here. Then this is one more diagram. This is one more picture. So here the efflux pumps. See, this is the normal bacterial cell. Here one efflux pump system is available. So what will happen when antibiotic, this red color dots indicates antibiotic, and antibiotic enters inside the cell, over accumulation of antibiotic occurs, thus makes the cell to die. Okay, but in certain organisms, that is antimicrobial resistant organisms, what will happen? Some mutation occurs, then some genes will be activated vigorously or they will be expressing overly, then thus trigger the expression of efflux pump system. In normal cell only one system is available. In resistant bacterial cell, see how many efflux system is available. So what will happen? These systems will be expelling the antibiotic outside. So there is no accumulation of required antibiotic. So because of that, what will happen? The organism will be releasing all the will be expelling all the antibiotic outside. Only lesser amount of antibiotic will be present inside. Thus makes the organism to survive. This is called antimicrobial resistance. Then quorum sensing. Quorum sensing, as I said already, it is nothing but bacterial communication. For communication purpose, the bacterial cells used to produce some signal chemicals. So through the signal chemicals, the resistant genes, that is one bacteria, come and join with another bacterial cells or one microorganism with another or a different microorganism, and they will be transferring their genes. That is called conjugation or transcription. Okay. So because of that, that is through the quorum sensing, the antimicrobial resistance can be spread bacterial population. Then, so what is the treatment? What is the solution we can go for this? In the sense, see here, solution in the sense we can go for patch therapy or phage therapy. It means nothing but by using virus we can kill bacterial cells. So if we want to kill pathogenic bacterium in humans or animals, we can give virus. So that virus should particularly kill only the bacterial pathogen. So that is somewhat the effective treatment introduced by the researchers, and it is somewhat successful. Then see the slide. After years of research, the scientists finding a new drug, and this new drug have been given for the treatment of a disease. So what the microbes used to say? New antibiotics. Oh, I got to evolve again. Then pharmacy company again. What pharmacy company will be doing? After years of research, they used to introduce new drug, it will be given for the treatment of disease. Again, the microorganism have to evolve again. So, antimicrobial resistance is one of the famous phenomenon, we can say famous phenomenon which occurs in microorganism very frequently. Then, these are the cause. So, conclusion, by patch therapy we can somewhat overcome the antimicrobial resistance, and these are the reasons that we have discussed in the previous slides itself, and that is improper use, adequate use, lack of education, lack of sanitation, poverty, and due to some other reasons the organisms gains antimicrobial resistance very frequently. So that's all about antimicrobial resistance. Thank you.